communities group students with common interests into the same floors or buildings. Virginia Tech has 13 living learning communities. Some are based on major, some on interest, leadership, or academic achievement. They're designed to put students in contact with people who share the same interests or goals so they can form strong relationships and help people build their own community within a big college.
I joined the LLC because I wanted to be a part of a community where everyone was of a similar major and took similar classes so we could bounce ideas off of each other and work together more fluidly than if went random.
In the Curie and Da Vinci community, every mentor group has the opportunity to challenge itself by taking on a semester-long project. These projects take many forms. Some are citizen science-based, others are more focused on building and design. For our project, we received Arabidopsis plants, some of which were wild-type and some that had altered genes. Arabidopsis is essentially a weed. You might find it growing next to the sidewalk in your neighborhood. It is a great study species because it grows quickly and has a short lifespan. So when you change something about its nature, the way it responds can be assessed easily.
So the purpose of citizen science experiments is so that scientists who are working on specific projects don't have to do all of the research themselves. They can crowdsource from other people not necessarily in that field, and then they do the research for them, and then the scientists can analyze it and know what's more important for them to look at and test out themselves in the future.
We were given some plants with an altered gene, and we knew that this particular gene affected photosynthesis. So we wanted to choose a variable that had something to do with photosynthesis because we thought that would likely give us meaningful results. We decided to look at phototropism, which is the ability of a plant to grow towards the light. We built a structure that suspended all the plant containers level but allowed the experimental ones to be tilted. Then the plants could grow for a period of time, and hopefully we would see them grow back towards the light. If that altered gene was affecting photosynthesis, it would make sense if those plants didn't grow back towards the light.
Arabidopsis is a really common plant, and scientists have completely sequenced the entire thing, so they know what all of the genes in the plant are already, but they don't yet know what each one does. So by testing the wild type versus the altered gene, we can help to figure out what the changed gene is, and then that will help us learn how that gene can affect other organisms.
So this is our experimental setup. We have 4 containers and they're all suspended using rope. And they're all hanging at 0 degrees relative to the light. So we let them grow for some time, and then we tilted them, the experimental groups, at 45 degrees. We let them grow for a week and then tilted them back, and then allowed them to grow for 1 more week.
This project has opened doors for us in the VT community by putting us in contact with several professors and giving us experience designing an experiment and carrying out in a lab setting. We got to work with faculty to get input and advice on our project and really gain experience that we can take with us when we look for a science-related internship or undergraduate research positions. And I really think that the overall purpose of the community is to get our names out there and to put us in a position to have a successful college career and to make an impact on the university.